Hello, and welcome to this introductory lecture in which we look at the question, what is embryology, and what it might mean for medicine in general. And the pattern we're going to follow is to look first of all at why embryology is important in medicine. An important part of that is looking at how common developmental defects are, and I'm going to use Western Europe as a reference frame because the frequency of defects varies from area to area. Next, we'll look at the early stages of development after fertilization, and in particular, the very early stages at which cells begin to become different from each other, because that process of differentiation is absolutely central to the process of understanding development in general and some of its medical implications. So looking at reasons why embryology is important, one valuable one for medical students is that anatomy often only really makes sense in the light of development. If you're looking at how the gut has developed, for instance, then that gives you an understanding as to why it's arranged in particular ways. Without that understanding, the anatomy becomes very hard to understand and very hard to remember. In addition, 2.4% of all babies born in Western Europe have a significant developmental defect, and that means one detectable at birth. If we were to follow babies forward, we'd find that about the same number again will show signs of a developmental defect that wasn't obvious at birth. Deafness sometimes falls into this category, or sometimes heart defects, which become evident when babies start to walk, putting increasing strain on the heart and showing up abnormalities that were not detected beforehand. So that means that perhaps as many as 5% of babies in total have a significant developmental defect, a number which is much higher than most people expect. Our next important aspect for medicine is reproductive technology. Things like in vitro fertilization and stem cell technology have revolutionized the early stages of assisted reproduction and look forward to having an even greater impact in the future. We can imagine, for instance, stem cells being used to grow an organ to replace an organ that in some way is not functioning properly, or to repair a spinal cord that's been damaged. Almost anything that we can think of might well be possible, but it's only possible if we understand the basics of human development. Then we find that many adult diseases, such as heart disease, cancer, even possibly schizophrenia, diabetes, have what I'm calling developmental antecedents, things that happened during the course of development that may well have influenced them. For some of these, we have good reason to believe that it's something that happens during the course of pregnancy, perhaps in the fetal period, that some influence has been exerted on the baby and that this will have consequences decades later for the adult. And if we understood what was happening here, then perhaps we would have a new tool to address many of these serious kinds of illness. Of these, cancer is probably the most significant as a killer of adult population in Western Europe. And in many ways, cancer cells are reverting. They're going back to embryonic behavior. They're behaving in the way that embryonic cells behaved in their past. And we'll look at that in more detail later within this lecture. So let's look at a bit of the natural history, what actually happens during the early stages in development. And this diagram shows an egg waiting to be fertilized. It's surrounded by cells which are derived from the mother. These are the corona radiata cells. And then by an impermeable membrane which does not contain cells itself. It's called the zona pellucida. There's a polar body which came from one of the meiotic divisions, and the nucleus is the only well-marked structure within the egg itself. And it's about 80 microns in diameter, which is small as eggs go. You've seen a hen's egg or an ostrich egg, so this is really rather tiny. But it's also large compared to the normal human cell. You can see the size of the corona cells nearby. That gives you an idea of what a normal human cell is like in terms of its size. So the egg is large for a human cell, small for an egg. It's also relatively featureless. The diagram doesn't make this quite clear, but if we look at a photograph of a newly fertilized human egg, you can see that the cytoplasm does not contain any obvious structures. It looks fairly homogeneous. And yet, in the correct environment, this single, rather small object is capable of turning into a full human being. So obviously, there are very many important changes that have taken place along the route, and it's some of these that we'll be looking at in this lecture. And you could call this the central enigma. How does something as apparently simple as an egg turn into something as apparently complex as a person? And you can see from the way I've phrased that question that perhaps the egg is not as simple as it looks, and perhaps, unexpectedly, people are not quite as complicated as you might think. Not everything is specified during the course of development. There are many examples where tissues interact with each other, they talk to each other, and come to an agreement about how they're going to be arranged in space.